खाना हम तो रात में हैं जमीन
Good evening, students. How are you all? All doing good? So, shall we start today with our new lecture? Today, I will not teach you so much. It will be lighter day. Let's have it light. So, let's start with gene expression from your syllabus of today's. Students, I might be taking an off on 22nd. So, today is 20th. So, on 22nd, so I will teach you tomorrow everything about that, or maybe I can try it today also. So that if I'm taking a holidays on 22, so your tasks are cleared before than that. So I will try that today. So we're dealing with the gene expression of your third syllabus, fifth unit one, third syllabus. So we will be dealing with that part today, gene expression and regulation of in eukaryotes. So let's start. So, gene regulation, it is a process of controlling which genes in a cell DNA are expressed. It's just a process to control your genes in a cell where DNAs are expressed. Different cells in a multicellular organisms may express different sets of genes even when they contain the same DNA. So in eukaryotes like humans, gene expression involves many steps. Gene regulation um, can occur at any of these steps. However, many genes are regulated primarily at the level of transcription. So let's start with the introduction of the gene regulation. So our bodies have different type of cells, from immune cells to skin cells to neurons. Almost all cells contain the same set of DNA instructions. So why do they work differently? Why they do have different jobs? The answer is gene regulation. It makes each cell different, these gene regulations. So gene regulation is how a cell control which genes out of many genes in a genome are turned on. Thanks to gene regulation, each cell type in our body has a different set of active genes. And these different patterns of gene expression cause various cell types to have different sets of proteins, making each cell type uniquely specialized to the job. For example, one of the job of the liver is to remove toxic substances like alcohol from bloodstream. To do this, liver cell expresses genes encoding subunits of an enzyme called alcohol dehydrogenase, and which breaks alcohol down into non-toxic molecule. And the neurons in a person doesn't remove toxins from the body, so they keep these genes unexpressed and turned off. Similarly, the cells of the liver don't send signals using neurotransmitters, so they keep neurotransmitter genes turned off. So here we can see that alcohol dehydrogenase in the liver cell, they are present and they are activated, leading to the production of RNA. Transcription is happening and alcohol dehydrogenase chain is activated. But neurotransmitter genes are deactivated because they are present in the brain. And these are your neurotransmitters. So these are present in your brain, and in the neurons, your alcohol dehydrogenase gene will be deactivated, but your neurotransmitter genes will be activated, and further doing the transcription by production of RNA. So they are activated in that case. So that's why your each cell type, liver cell, brain cell, they're acting differently. They have different roles. They have different functions. They have different production of proteins. They have different level of set of working out things. So there are many other genes that express differently between liver cells and neurons. So they act differently. So how do cells decide which turns to turn on? How do they decide that which gene has to be turned on or turn off? So many factors affect different genes to express in our body. Different cell types express different set of genes, as we have seen above. So however, two different cells of the same types may also have different gene expressions depending upon the environment internal state. So from the same cell type, the two different cells might be uh, of the same cell type. Two different cells might have different expression patterns because of the environmental and internal state. So gene expression could be determined information from both inside and outside the cell. Example of information from inside the cell: protein is inherited from its mother cell, whether its DNA is damaged or how much ATP it has. So example of information from outside the cell: chemical signals from the other cells, mechanical signals, extracellular matrix, nutrient levels. So all are the example of your outside cells. Insides are the proteins which are inherited from the mother cells, whether DNA is damaged and how much ATP it has. So how do they queue up whether these genes has to be expressed or not? So cells don't make decisions to sense that you should or I should would. So they have molecular pathways convert information such as binding of chemical signals to its receptors into a change in the gene expression. So they get binded to a chemical signal into a receptor and change into gene expression. So as an example, let's consider how cell responds to growth factors. A growth factor is a chemical signal from the neighboring cell that interacts the cells to grow and divide. So, so here we can see that there's a growth factor attaching to the receptor. It's activating various cell types, various proteins, which are attaching to the transcription factors, leading to division promotion gene, leading to growth promotion gene, which are further producing in the translation step your proteins that drive growth and division. So that's how your whole this system works out. From your receptor accepting the growth factor, then in transcription factors are activated, and then the translation is done per leading to production of proteins that which growth and division has to take place. So the cell detects the growth factors through physical binding of the growth factors to the receptors on the cell surface, and binding of the growth factors causes the receptor to change shape, triggering a series of chemical events in the cell that activate proteins called transcription factors. So further, these transcription factors bind to certain sequence of DNA in the nucleus, causing transcription of cell division. And further, the products of these genes are of various cell types, making the cell divide further. So they are just one example how cell can convert a source of information into change into gene expression. So there are many other things that are happening at the biological level today. So growth factor signaling is a complex involving the activation of a variety of targets, including both transcription and non-transcription factor proteins. So eukaryotic gene expressions can be regulated at many stages. So in the eukaryotic gene regulation, we know that how the expression of gene in eukaryotes can be controlled at various stages, whether availability of DNA, production of mRNA to the translation and processing of proteins. So during this whole case, different genes are regulated at different points, and it is not uncommon for genes to be regulated multi-steps. So at the level of chromatin step accessibility, the structure of chromatin can be regulated. More open or relaxed chromatin makes a gene more available for transcription. At the stage of transcription is a key regulatory point for many genes. Set of transcription factors, proteins bind to specific DNA sequence or near gene, represses transcription into RNA. RNA processing, splicing, capping, addition of poly A tail to an RNA molecule can be regulated and can exit from the nucleus, and hence could lead to the some pre-mRNA by alternative splicing. So this is how your eukaryotic gene expression works like. So you have a chromatin structure. The structure, your chromatins are removed, your histones are removed. Then you have a transcription leading to processing of mRNA, and it's going out of the nucleus. Then it's being attached by the ribosomes and tRNA, leading to the translation processing happening. And then you have active protein, and there's various post-translational modifications happen in your protein to work for its desired state. It's also depend RNA stability. The lifetime of mRNA molecule in the cytosol affects how many proteins can be made from it. So small regulatory RNA called microRNAs can bind to target mRNA and cause them to chopped up. At the translation level, translation of mRNA may be increased or inhibited by the regulators. For instance, microRNA sometimes block translation of the target mRNA. Protein activity can undergo a variety of modifications, such as being chopped or tagged up with the chemical groups, and these modifications can regulate it may affect the activity of behavior of the protein. So that was short and indeed uh, simple to understand, nothing much. Now comes the interesting part, that is Lac Operon and fine structure of genes. So, so we understood now gene expression, how gene expression works, various gene activations and various processes in a short and nutshell. So now comes the topic of your Lac Operon and fine structure of gene. So here we will discuss how Lac Operon, which is important for the glucose and lactose operation in the case of gene activation, and also we will deal with the TRP trip operon also, how their functions are, what are their various paths are, how they are important, and how it works out. So we will deal with all these parts today with this Lac Operon and fine structure of gene. So bacterial genes are often found in operons. Genes in an operon are transcribed as a group and having a single promoter. And in each operon contains a regulatory DNA sequence which act as a binding sites for regulatory proteins that promote or inhibit transcription. So regulatory proteins often bind to small molecules, can make the protein active or inactive by changing its ability to bind DNA. And some operons are inducible, meaning that they can turned on by the process of a particular small molecule. Other are repressible, meaning that they are on by default, can be turned off by small molecules. So how gene expression is regulated, as we have discussed already, there are various forms of gene regulation. The controlling is done by the gene, they get expressed at which level. However, a lot of things are happening at the level of transcription. So like in this case, in bacteria, they have example of given to you bacterial chromosome inside there, and inside the bacterial chromosome, this is the operon. This is the operon structure is made up of. Operon is mainly has promoter where RNA polymerase binds, and it has gene 1, gene 2, and gene 3. uh, and these genes works together, all these genes, and then the transcription happens. Then you have mRNA with gene 1, gene 2, and gene 3, leading to the production of protein 1, protein 2, and protein 3. Hence you have uh, yeah your gene regulation in bacteria works like this. So in bacteria, related genes are often found in cluster of chromosomes where they are transcribed from one promoter as a single unit. Such cluster of genes under control of single promoter known as operon. And operon are common in bacteria, but they are rare in eukaryotic such as humans. So in general, we will contains an operon will contain gene that function in the same process. So we will study the Lac operon here, that contains gene encode the various involved uptake and metabolism of particular sugar, lactose. Operon allows the cell to efficiently express set of genes produced when needed at the time. So this is how your operon looks like. So you have a promoter, you have gene 1, 2 and 3, and whenever the RNA polymerase is attached to the promoter, transcription happens. But in some of the cases, operons also have addition to the promoter, there is the operator, and and the operator, mainly the repressor, are attached. Some regulatory proteins are repressor that bind to the pieces of DNA called operators, and when bound to operator, repressor reduces the transcription. So it's where it doesn't let the RNA polymerase to be attached to the promoter and leading to the less transcription and has no mRNA made. So no mRNA is made and leading to this kind of situation. But sometimes they are activator also, activator on the left side. When they bind, they touch to they activate the polymerase, RNA polymerase, bind to the promoter, and lots of transcription happen and lots of mRNA is made. So that is also is happening in our operon case. So there is activator, promoter, then there is three genes which are doing lots of transcription with the help of RNA polymerase. And if at the operator repressor is attached, then your promoter will not work, and but in the other case, yes, it will work. So how the regulatory genes works out? So and these genes encode regulatory proteins are sometimes called regulatory genes, and many regulatory proteins that can themselves turned on or off by specific small molecules, and the small molecules are bind to the protein, changing its shape, altering its ability to bind DNA. For instance, an activator may only become active when it is attached to certain small molecules. So whenever activator is off, uh, so you have no binding, so nothing will happen. But when you have a small molecules attached to it, then you have activator is on, then it binds to the target DNA. Only then it could do the functions of it. Okay. And some operons are usually off but can be turned on by small molecules. The molecules are called inducers, and operon is said to be inducible in that case. So there are two types of operons, Lac Operon and Trip Operon. The Lac Operon is an inducible operon that encodes enzymes for metabolism of sugar lactose. So it turns only on when the sugar lactose is present, and the inducer in the case is allolactose, a modified form of lactose. Others operons are on but can be turned off by small molecules. The molecules are called co-repressor, and operon is said to be repressible. Example is trip operon, TRP operon is a repressible operon that encodes enzyme for synthesis of amino acids tryptophan. So let's start with the Lac operon now. How its structure is, how it works out, how's the mechanism behind the Lac operon is. So, Lac operon of E. coli contains genes involved in lactose metabolism. It's expressed only when lactose is present and glucose is absent. So, whenever lactose is present and glucose is absent, only then Lac operon works out. So, two regulators turn the operon on and off in response to lactose and glucose level. The Lac repressor and Catabolite activator protein. So, there's a Lac repressor there which act as a lactose sensor. It normally blocks the transcription of the operon but stops the acting as a pressor when lactose is present. So it sends the lactose indirectly through its isomer allolactose hormone isomer. Whereas a catabolite activator protein CAP acts as a glucose sensor, it activates transcription of operon but only glucose levels are low. So CAP senses glucose indirectly through the hunger signals molecule from CMP. So let's see them each of them carefully. So here you can see the Lac operon has three genes, Lac Z, Y, and A, and these genes are transcribed as a single mRNA under the control of one promoter. So genes in the Lac operon specify proteins that help utilize the lactose. So like Z gene encodes enzyme that splits lactose into the monosaccharides that can be fed into glycolysis. Like Y genes encodes a membrane embedded transporter that helps bring the lactose into the cell. And in addition to three genes, Lac Operon also contains regulatory DNA sequences. These are the regions of DNA to which particular regulatory proteins can bind, control transcription of the operon. So the Lac Operon here it looks like cap site, operator, Lac Z, Lac Y, Lac A. So here your promoter cap site will attach to this site. Here the RNA polymerase will attached. Here the repressor will attached. So repressor will leads to the node transcription, whereas the promoter region, RNA polymerase binding region with the cap, it will leads the positive feedback. So that's how your Lac Operon, the major anatomy, looks like. So the promoter is binding site for RNA polymerase enzymes that perform transcription. The operator is a negative regulator site bound by the Lac repressor protein, and the cap binding site is a positive regulatory site that is bound by the catabolite activator protein. Let's take a closer look how they works and how their things are working out. So here we can see when lactose is not present, in that case lactose is absent, so Lac repressor binds tightly to the operator. It binds to the operator tightly. It gets DNA, it's get onto the way of RNA polymerase way, preventing that transcription. It does not let transcription happen. But when lactose is present, allolactose binds to the Lac repressor and it makes it to let go of the operator, and RNA polymerase cannot transcribe the operator. So because lactose is present, because of that allolactase binds to the Lac oppressor. This allolactose binds to the oppressor, letting the RNA polymerase attach to the promoter and letting the transcription happen in common way. So that's how the lactose with no lactose and with lactose are so important in this case. So now comes the part of CAP, how CAP is working. CAP workings in two cases with it low glucose and high glucose. When there is a low glucose levels are there, CMP is produced, your cyclic adenosine monophosphate, and which attaches to the cap and attached allowing it to bind to the DNA, and cap helps uh, RNA polymerase binds to the promoter resulting in high level of transcription. So whenever there is low glucose, cap with the help of CMP, cap will attach to the cap site letting the RNA polymerase attach to the promoter, leading to the production of your high transcription of your uh, lac protein. Whenever there is high glucose, so no CMP is produced, so cap cannot bind to DNA without CMP, so transcription occurs only at very low level in this case. So that's why we have less transcription because CMP cannot be attached to it. CMP cannot attach and cap cannot attach. So that's how low glucose, high glucose level works here. Students, this is one of the toughest and complex system to understand, but with these diagrams, they are quite easy to understand, and I try to simplify things as much as possible for you. So we are just concentrating on them, so that tomorrow we will start with the ultrastructure of cells, which you might know. So I will not go through details, but I will take care of your practical parts. We will take some virtual labs tomorrow, and then yeah. So just concentrate on this part today. It will be light today for you, all of you. Not much of your things. So so when does the Lac Operon really works? When does it turn on? Mainly in two conditions. When glucose must be unavailable, glucose is unavailable. CMP binds to CAP, making CAP bindable to DNA, and leading to the Lac Operon promoter attached. And lactose must be available. If lactose is available, the Lac repressor will be released from the operator by binding of the allolactose, and this allows RNA polymerase to move forward on DNA and transcribe them. So the two events in combination, if these two events happen, then you have a strong production of your mRNA in that case. So this is a summary, that is glucose present, lactose absent. So no transcription of Lac Operon occurs. That's because Lac repressor remains bound to the operator and prevent the transcription by the RNA polymerase. Also CAMP levels are low because glucose levels are high, so CAP is inactivated and cannot bind DNA in that case. So repressor is attached in this case, glucose is present, lactose is absent, so it's uh, no, very bad for production of RNA. But in the case of glucose present, lactose present, so low level of transcription will happen. Uh, but Lac repressor is released by attaching of allolactose. So RNA polymerase will produce, but glucose present, because of that cap cannot attach to the cap site there. But when glucose absent, lactose is absent, so cap could be attached, but a repressor will be attached to the operator. So this will lead to the also not so roadblock to RNA polymerase, and it will prevent transcription. This is the best part when you have glucose absent, lactose present. So glucose is absent, then CMP could be attached and cap and cap side, and also allolactose will attach to repressor. So you have high amount of transcription is happening in this case. So this is example in summary. So you have glucose present, lactose absent, repressor binds, cap does not bind, no transcription. Then low level transcription, no transcription, strong transcription. So this is a summary of all the parts that we have done. Now comes your second type of operon. It's not included in your syllabus, but we're still doing it. It's quite interesting to go with this also. So tripoperon found in E. coli bacteria is a group of genes that encode biosynthetic enzyme amino acid tryptophan. So tripoperon is expressed when tryptophan levels are low, repressed, and when they are high. Tryptophan is located by the repressor genes. When bound to tryptophan, tryptophan blocks expression of the protein. So here, trip proteins, we can check it here. So it has five different genes, sorry, trip E, trip D, trip C, trip B, trip A, and here your operator is attached, and also then after the transcription, mRNA is produced, then translation, enzymes for tryptophan biosynthesis are produced. So this is how your trip operon usually looks like. It also have repressor at the operator side where tryptophan is attached to it, and leading to not to whenever there is high tryptophan, it will attach to the tryptophan repressor, it will be activated, and you will have no transcription in that case. But in case of low tryptophan, your tryptophan repressor will be inactivated, your promoter RNA polymerase will be attached, and transcription could be happen like this. So that's the main thing with the tryptophan and Lac Operon. That was the lecture today, students. It was not much for today, so I will share these lectures with you soon. And yeah, that's it for today. Not much, because if I start teaching you today about a study of chromosome and structures, I want to teach you this part also. Would you like to learn about in the next 30 minutes about techniques from MBBT? That is protein isolation. That is also quite important in whenever you're working with the genetics work. So you have to isolate protein and you should be aware how it is done. Shall I play that videos for you today, or we just done for today? You can relax and we see us tomorrow with continuity. Tell me how you feel like I can continue in that way. Because this part could be done in 1 day, and then this one and this one in 2 days. So this is not much left for us. So mainly these were the many topics that we have to initiate in the start, and then slowly and slowly we will deal with these parts also. So it's not much for today. Which video, बच्चे? You mean for the protein isolation? Mm, or we can go. Okay, I will share here only. Just wait a second. So students, please subscribe. Come to my YouTube channel. So here you can see all types of videos that you want to get through. So I think in the last lectures, so here you can see my principle of genetics, all the lectures coming up, principal plant biotechnology, MBBT, if somebody want interest in the NEET, so everything is there. So just subscribe to the channel so you can have all the values that you're looking for. So yeah, that was the link. I can also teach you if you want this part. So this is the link for the techniques. So I can start then. We have 30 minutes. So in meantime, I send you today's lectures. Okay, in WhatsApp. No, I think it's enough. Just go to my WhatsApp, to my YouTube channel. Just from the 10th video, you can find it there. So I will also put what kind of techniques you can learn from there. So don't worry. Okay, I have to now look for my workshop. So everyone is there. Okay. So that was a workshop by MBBT. So I'm teaching these techniques also. These techniques I will be having a lot of videos, a lot of interesting things that will be required in your genetics part also as an experiments. So let's start with the protein isolation and quantification and purification. So it will be divided into different assays: homogenization, fractionations, centrifugations, quantifications, and chromatography, and then electrophoresis. So these parts will be divided for this workshop. So these are the various Nobel Prize winners on the chromatography and electrophoresis. Uh, sorry, Archer John Porter Martin, Richard Lawrence Millington Singh, and Cora Stilston. So they did their work in the field of partition chromatography for the invention. And then mass spectrometry sedimentation. John B. Fenn, Tanaka, and Swedberg, they work really good in the field of mass spectrometry. Then to separate your proteins by organelle fractionation, Albert Claude, Christian de Duve, George E. Pallet. So these are the various people who worked really hard and they got the Nobel Prize winner for their work, what they did. Then on the protein sequences, Frederick Sanger and Frederick Sanger both worked in United Kingdom in the Cambridge for the contribution of the structure of proteins, especially that of insulin, and for the contribution on the base sequence in nucleic acids, respectively. So these are various assays that you are seeing. That biochemical or biological means used to monitor the presence of target proteins. For example, enzyme activity, specific labels, functional proteins such as bindings. So here you have a lactate and pyruvate. Lactate dehydrogenase is being added as an enzyme in that case, where it changes your structure uh, into pyruvate. So this is how you can check with the help of enzyme how much pyruvate is produced by addition of enzyme into that. And same hold for the spermidine and deoxypyrazine synthase. You can check the volume of your deoxy isopuzyl E alpha or hypopuzyl E alpha A. Then how you can isolate protein? You can isolate proteins by two different methods, that is homogenizations and fractionation. In homogenization, first step is to disrupt your cells. Then in fractionations, crude separation of homogenes into fractions. So homogenization could be done by Downs homogenizer, sonication, osmotic shock, or French press. And fractionation has to be taken care with the help of pH and heat, ammonium sulfate precipitation, differential centrifugations. All these things are being taken care in that case. So centrifugations at 500 G for 10 minutes. Uh, then homogenates are formed. Then you have a supranatant, and in the palette you have first nuclear fractionations. Your nuclear material is there. Then you again centrifuge it for 10,000 G for 20 minutes. Then you find mitochondrial fractionations. Then again for 100,000 G, then you find microsomal fractionation. So it keeps on going deeper and deeper the more you go for fractionation of your mixture of cells. Then comes your protein isolation. Some other part by centrifugation at different molecules, as macromolecules are based on density and shape. You can separate each of them as per your requirements. So here you can see differential centrifugation. Sample are poured into tube, more dense materials are produced, and you can do it with the differential centrifugation and rate zonal centrifugation. So in the differential, you just put it in there. So your particles settle down according to the mass. Then you centrifuge. Then you stop the centrifuge. Then you check the palette. Then you have decant the liquid into container. And then yeah, so you have your at the bottom your cells that you're looking for in the palette. Rate zonal centrifugation, sample is layered on the top, large particles, small particles. Sucrose is the gradient. You have a centrifuge, and then you have two different gradients, and you can remove them one by one according to the increasing mass of particles, like this. So protein isolation centrifugation is done at various levels: 3000 G to 110,000 G to 40,000 G, 200,000 G, 4,000 G, and you are getting various materials. So like large organelles, mitochondria, membranes, plasma membrane, large protein follicles, and proteins, etc. So this sedimentation is depending upon the sedimentation coefficient, which is equivalent to the 10 is to power minus 13 seconds. So that's one formula for the same. So this is how the same method that we were discussing before, with the sedimentation coefficient, your all different protein particles, they are separated as per uh, the molecules that you're looking for. So this is the various values for Swedberg units for different part of your cells, and this is the density of sedimentation coefficient graph, where you can have a look for various molecules as per the sedimentation coefficients. So how you can check then? One thing is done, that you have extracted, you isolated your protein. Now it's come the chance of quantification of protein. This could be done with the help of Bradford assay. So this technique is simpler, faster than the Lowry method, is subject to less interference. In this case, a Comessi blue G250 dye appears to bind mostly readily to organyl and lysyl residues of proteins, and this specificity can lead to the variation in the response of the assay to different proteins, which is the main advantage of the method. So let's watch video.
Protein quantitation is one. In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through, and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample, and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. . Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample. Estimate the protein concentration of the milk sample based upon the visual comparison. To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
So that was about Bradford assay. Now comes the ultrasensitive fluorescence based protein quantitation. So in this case, you can quantify your proteins by adding a dye to samples. You heat your sample for 10 minutes, cool to room temperature, then there will be some fluorescence which you can check it by the spectrophotometer. Then you can have this range of fluorescence versus protein like this, and then you can check it your results. So technique, then comes the protein purifications techniques, so such as dialysis technique used to remove small molecules from the protein solutions. So here you can see there is small and large molecules. So at start of dialysis and at the equilibrium, your large molecules remain inside and the small molecules are outside. So these are the protein Proteoplus kit. It contains proprietary buffer and precipitation protocols for preparing samples for the matrix assisted laser multi-MS cryobiology. So this is, let's watch it. Hope that was clear how it is done. Then liquid chromatography is a term which refers to the chromatic method in which mobile phase is liquid and stationary phase may be liquid or solid. So here you have a different beads to which your small particles like red ones, they got attached inside of your beads, and the green one also attached but not so much, and the yellow ones got separated. And then based on that you separate your yellow, green and yellow beads by the different level of the separations out of your samples. So this is gel filtration chromatography. So we're going to talk about in the gel filtration chromatography, your layer samples are on columns. So you have large proteins and small proteins. Large proteins will just flow out, and the small proteins will get attached to the uh, to the beads, and then later on with the help of fractionation they can be also removed. So like we have a gel called Cephadex G75, Cephadex G10. You can separate them like that. So let's watch.
We're starting with gel filtration medium. This is Cephadex G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins above 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In the speaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now I'm going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. So this is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom. Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of 2 molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1 million. Fluorescein, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these 2 molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and I've made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescein, 400 microliters of the blue dextran. Nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I'll hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in, so I'm adding buffer very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first to help get everything into the gel before I add a large volume of buffer. So now the separation has proceeded somewhat. I'm going to try to add a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on the top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50 and they're starting to separate quite nicely. The yellow molecules are up here, the blue are down here. Still got a little overlap. So I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. Fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9. About 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely. And what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the dextran that's in the column if you're storing it over a long period of time.
So that was quite simple and self-explanatory video about what we have discussed here, how you can separate a heavy molecule and with a small molecule by gel filtration Cephadex S50. Now comes next chromatography techniques, that is ion exchange chromatography. In this, protein in a solution that has low ionic strength is added to the column, and protein interacts with the beads via electrostatic interaction. So like here, the one with the positive charge, they got interacted, and the one with the negative charge, they got eluted out. So here you can do it in this way. So there could be an ion exchange chromatography where matrix carries a net of positive charge which interacts the negatively charged protein strongly. So, and in the cation exchange, the matrix is negatively charged and interacts with the positively charged proteins. So this is a cationic exchange chromatography. So let's watch one video for this also.
Chromatography is a chromatography process that allows the separation of ions and polar molecules based on their charge. As well as other forms of chromatography, in ion chromatography, there is a mobile phase, which generally consists of buffer solution. And there is a stationary phase which consists of a matrix that contains charged ionizable functional groups. This type of chromatography is subdivided into anion exchange chromatography and cation exchange chromatography. Anion exchange chromatography. Negatively charged molecules are attracted to a positively charged solid support, which is usually beads that provide a positively charged surface, such as resin quaternary ammonium. In cation exchange chromatography, positively charged molecules are attracted to negatively charged solid support, which is commonly beads that provide negatively charged functional groups, such as resin methyl sulfate. Ion exchange chromatography is frequently used for the separation and purification of proteins. The basic process of this chromatography can be represented in 4 steps: conditioning or equilibration of the stationary phase, sample loading, washing, and elution of the retained molecules. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and ionic strength of the sample buffer are selected to ensure that, when sample is loaded, proteins of interest bind to the medium, and as many impurities as possible do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached, all stationary phase charged groups are bound with exchangeable counterions, such as chloride or sodium. When the column is equilibrated, the second step is sample application. Depending on the pH of their environment, proteins may carry a net positive charge, a net negative charge, or no charge. This net charge can be determined by comparing the pHi of each protein with the pH of the buffer. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each amino acid. Depending on the pH of their environment, the amino group can be protonated and become positively charged. The carboxyl group can be deprotonated and become negatively charged. And the side chain of some amino acids can become positively or negatively charged due to the gain or loss of protons. So, the net charge on the molecule can be positive, neutral, or negative. The isoelectric point, or pHi, is the pH at which a protein carries neutral charge. At a pH below their isoelectric point, proteins carry a net positive charge. And at a pH above their isoelectric point, they carry a net negative charge. In this example, the pH of the buffer is equal 5, consequently, protein A has a net negative charge. And both protein B and C carry a net positive charge. In addition, protein C is more positively charged since its pHi is higher than the pHi of protein B. If we apply cation exchange chromatography, protein that has a net positive charge, it will bind to the negative charge beads of the stationary phase. On the other hand, uncharged proteins, or those with a net negative charge, would not bind. They pass through the column at the same speed as the flow of buffer. When all the sample has been loaded, the next step is column washing. The column is washed with start buffer. To ensure that all non-binding proteins have passed through the column. When all the sample has been loaded and the column washed, conditions are altered in order to elute the bound proteins. Most frequently, proteins are eluted by increasing the ionic strength of the buffer, or, occasionally, by changing the pH. As ionic strength increases, the salt ions compete with the bound components for charges on the surface of the medium, and one or more of the bound species begin to elute and move down the column. The proteins with the lowest net charge at the selected pH will be the first ones eluted from the column as ionic strength increases. Similarly, the proteins with the highest charge will be most strongly retained and will be eluted last. Protein B has the lowest net positive charge, so it can be eluted first. Then, protein C with the highest net positive charge, it can be eluted next, by increasing the salt concentration. Also varying the pH of the buffer can be used to effect a separation, and to elute the retained proteins. As we seen, the pHi of the protein B is equal 7, and the pHi of protein C is equal 9. So if we add a buffer with pH above the isoelectric point of protein B, and below the isoelectric point of protein C, the protein B will become negatively charged, and can then be eluted. The protein C is still charged positively, consequently, still retained. Next, protein C can be eluted if we apply a buffer with pH above its isoelectric point. So, it becomes negatively charged, and it will pass through the column. One of the primary advantages for the use of ion chromatography is only one interaction involved during the separation. Another advantage of ion exchange is the predictability of elution patterns. By controlling changes in ionic strength or in pH of the buffer, using different forms of gradient, proteins are eluted differently in a purified, concentrated form.
So, बच्चों, that was about electron ion exchange chromatography. I think uh, I hope that will be enough for you just to have a glimpse of it. Would you like to continue, or you're still with me? Because I can see only 9 concurrent views so far, and most have gone for Sunday fun day. How many you are still there? Could you confirm with me? Yes, with a thumbs up. Okay. Okay, one reply is there. It's okay. So even though there is one student, I will teach. So I will continue. Then comes the affinity. This is based on the antibody based. So you have antibody that is glucose binding attached to the glucose, and you have glucose binding on the released on the addition of glucose. So here your primary antibody attached to your beads, and then comes your secondary antibody, and then secondary antibody doesn't attach, then it's washed away, and you're gonna elute it with a different pH actually. So drawbacks are this: the binding to the ligands or antibody can often be strong, that very harsh conditions are needed to elute the enzymes. This leads to the denaturations. Non-specific binding may also occur, specifically there where large ligands or antibodies are employed. Thus, there is often a limit to the protein's and concentration that can be applied to affinity column. So
In affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and elution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
So that was about affinity oh, it's hot. Affinity chromatography. Then comes the chromatography, HPLC. That is high pressure liquid chromatography, in which you can separate your molecules on the basis of the columns. So these are the various peaks you can see of your things. So I will just start this one.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase non-polar. In paper chromatography, the solvent moves along the paper by capillary action. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention.
The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
So the HPLC, that's I want to show you at the end, because this is the highly recommended technique to isolate your protein, to purify your protein. And on that note, we are done with the protein purifications today. I think that's it for today. And you can check in that link if you're looking for that I've shared with you today. So for today we are good now. I wish you all good night. Okay, what is the most common procedure? Yeah, it was HPLC. It was. So it was HPLC. So I wish you all a good night. Take care. See you tomorrow, same time, same place. Till then, keep smiling. Bye bye.